Welcome to Module 3, Stem Cell Differentiation. In this module, you will learn the main purpose of adult stem cells and how adult stem cells are activated in order to perform this function. The main purpose of adult stem cells is to help repair and maintain various tissues in the body. They do this by a process called differentiation. Differentiation is the process where a stem cell specializes or develops into another type of cell. But in your body, you have a limited number of adult stem cells available. In order for the number of adult stem cells to remain the same, they undergo asymmetric division. Which is when a stem cell divides into one identical daughter cell and one differentiated cell called a progenitor cell. A progenitor cell is like a stem cell in that it can differentiate into another type of cell, but it is different in that it is already on its way to become a specific type of cell. In order for progenitor cells to repair tissue, they must also proliferate by symmetric cell division. Wherein a cell divides into two identical daughter cells. This is also called progenitor division, shown twice here. Cells then undergo terminal differentiation. In order to become the target cell, which has a specific function within the organism and will proliferate as only one cell type. These cells express genes that are specific to their cell type and function. Differentiation depends on cell signaling. In which stem cells receive signals that are translated into transcription factors that alter the expression of the DNA contained within them. This causes stem cells to undergo an irreversible change and differentiate into a more specific type of cell. An example of this is the protein noggin. Which induces mesenchymal stem cells, or MSCs, to become adipocytes. An example of a differentiation tree of an adult stem cell is shown here, where an MSC undergoes symmetric cell division and then differentiates into progenitor cells. Through osteogenesis, the MSC becomes an osteocyte, which then differentiates into bone tissue. Through chondrogenesis, the MSC becomes a chondrocyte, which then differentiates into cartilage tissue. Through myogenesis, the MSC becomes a myoblast, which then in turn becomes muscle tissue. Through tendogenesis, the MSC becomes a fibroblast, which then differentiates into tendon tissue. Through adipogenesis, the MSC becomes an adipocyte, which then in turn differentiates into adipose tissue.